So next we're going to hear from one of our previous ESPID Fellowship Award winners. The ESPID Fellowship is our most prestigious award that we make to support people undertaking research, and we usually ask one or two previous fellows to return each year to tell us about what they've done with the ESPID money that they received. Usually it's very exciting to hear the results, and this year we're going to hear from Ioanna Papadatou, who's going to tell us about systems analysis of immunity following pneumococcal vaccination.
Thank you, Aubrey. Good day. Before I begin, I would like to thank the board and the committee for this fellowship that has really enabled me to promote my research on vaccine immunology. So nothing to disclose. And a little bit of background. I'm a pediatric infectious diseases fellow and a postdoctoral researcher in the University of Athens in Greece. And my work focuses on vaccine responses in high-risk individuals. What we know is that preterm infants are at high risk for serious infections, and at the same time, it is well established that they mount low antibody responses to pneumococcal immunization compared to healthy term infants. So my question was, what does this mean for their long-term immunity against pneumococcus? Because as you know, long-term protection conferred by a vaccine relies not only on the initial antibody response, but also on the continuous secretion of antibodies from long-lived plasma cells from the bone marrow, and most importantly, at least for me, on the memory B cells that they are formed and have the potential to generate robust secondary responses upon rechallenge. And in this study, we will discuss two main phenotypes of memory B cells. The classical MBCs that derive from germinal center reactions are long-lived and have high affinity, and the non-classical MBCs that are born outside the germinal centers and are thought to have lower longevity and affinity. And of course, today, we're all learning that all these immunological processes are orchestrated by a cascade of transcriptional signatures. And from previous studies, we knew that age has a crucial role on the nature of memory B cell response to pneumococcal immunization, with extragerminal non-classical MBCs dominating the response in younger infants compared to older children. And also from the literature, we have learned that age plays a crucial role also in the transcriptional profile of the immune response to vaccines, for adults and for children the same. So we designed this study with the help of the ESPID Fellowship in order to compare the transcriptional profile, the memory B cell, and the antibody response to a 3+1 immunization schedule with a 30-valent conjugate vaccine in preterm and full-term infants. And for the gene expression, we used whole RNA sequencing that was then functionally annotated using bioinformatic tools. And for the enumeration of the pneumococcal serotype-specific MBCs, we have developed a method in our lab that you can find online the details, but basically it enables us to better detect rare events such as the antigen-specific cells. So what we found is that primary immunization with PCV13, the third dose of the vaccine, resulted in a much stronger gene perturbation in preterm infants compared to full-term infants, with 265 differentially expressed genes in the preterm cohort in comparison to the mere 29 in full-term infants. And in order to study the functional properties of these signatures, we used gene set enrichment analysis and string analysis. And what you can see is that preterm infants mounted a robust innate response marked by the expression of pro-inflammatory signatures. And this was also confirmed when we directly compare the two groups, preterm versus full-term infants, in the post-immunization time point on day 2 after the third dose of PCV13. And we found that interferon type 1 signaling was significantly upregulated in the preterm infants compared to the full-term infants. And as far as the memory B cell response is concerned, we found that memory B cells against the serotype tested for serotype 1 and 9V increased significantly in both groups, and at 1 month post-immunization, there was no significant difference in the absolute count of the cells. However, as expected, when we looked at the subphenotypes, the extragerminal non-classical MMBCs were much higher in the younger, the preterm infants. So, in summary, we have shown that preterm and full-term infants mount distinct transcriptional and cellular responses to pneumococcal immunization, with the preterm infants demonstrating a more robust pro-inflammatory response to immunization, but at the same time, a more immature memory B cell response derived mostly from outside the germinal centers. And altogether, our findings emphasize the role of prematurity in the shaping of the immune response and the need for tailored immunization schedules for this high-risk group. Finally, I would like to thank the many people who work on this project, the neonatologists, immunologists, and bioinformaticians, and special thanks to my team, Irene Jovara and Mary Giropepa, and of course our mentor, Vanna Spoulou. Thank you.